Hi, in this video, I will talk about membrane potential, action potential, and chemical synapses. Nerve cells are considered electrically excitable cells because they can generate signals called action potential, also known as nerve impulses, in response to change in plasma membrane voltage initiated by stimuli. In order to understand excitability of the nerve cell and how it leads to action potential, let's first look at the anatomy of the nerve cell. Nerve cell, just like other cells, is made up of cell body, also known as soma. Within the cell body, you will find cytoplasm and cellular organelles such as nucleus. The cell body has extensions referred to as processes. One type of processes are called dendrites. Dendrites are highly branched and are responsible for receiving most of the electrical signal information from other cells. Another type of process is called an axon. The cell body, as well as the processes, are surrounded by plasma membrane, composed of phospholipid bilayer. The function of plasma membrane is to regulate what enters the cell and what leaves the cell. Because plasma membrane is composed of phospholipids, only hydrophobic and non-charged substances can pass directly across plasma membrane. Charged particles, such as sodium or potassium ions, require channels to enter or to exit cell across plasma membrane. When you look at the distribution of these ions across plasma membrane, you see that there is higher concentration of positively charged sodium ions and negatively charged chlorine ions outside of the cell than inside of the cell. And concentration of positively charged potassium is higher inside of the cell than outside of the cell. The difference in the concentration of these ions on opposite sides of the cell membrane creates membrane potential, which is measured in millivolts. Specifically, with the ion distributed across the membrane at this concentration, the resting membrane potential is at minus 70 millivolts. Keep in mind that resting membrane potential describes steady state of cell when there is no action potential. Now, to initiate action potential, the membrane potential has to change. This starts with channel opening for sodium ion in the membrane. Because the concentration of sodium is higher outside of the cell than inside of the cell, sodium ion will rush into the cell once the channel opens. Because sodium is positively charged ion, it will change the voltage inside of the cell from negative 70 millivolts to positive 30 millivolts. The change in voltage from minus 70 to plus 30 millivolts is referred to as depolarization. As the membrane potential reaches positive 30 millivolts, channel for sodium closes and channel for potassium opens up. Because there's higher concentration of potassium inside of the cell than outside of the cell, potassium leaves the cell, taking a positive charge with it, and the membrane potential begins to change back towards negative 70 millivolts. This is called repolarization, meaning that membrane voltage moves back towards the resting membrane potential. However, because potassium channels are slightly delayed in closing, the voltage drops below negative 70 millivolts before returning to negative 70 millivolts. This is called hyperpolarization. After that, sodium-potassium ATPase pump and other non-gated channels allow sodium to return outside of the cell and potassium to return inside of the cell. What has been described here is the action potential, which is by definition rapid rise and fall in voltage cellular membrane. This action potential can be represented by the graph of voltage over time. The membrane potential will stay at the resting voltage, which is negative 70 millivolts, until physical stimulus triggers or activates sensory receptors in dendrites of a sensory neurons. For example, when someone touches you, stimulus triggers pressure receptors of sensory neurons located in your skin. This induces sodium channels to open, allowing sodium to enter the cell. Due to increase in positive charge inside of the cell, membrane potential changes from negative 70 millivolts to 0, and then to positive 30 millivolts. This is depolarization. At positive 30 millivolts, or the peak of action potential, sodium channels close and potassium channels open up, allowing potassium to diffuse outside of the cell, causing membrane potential to return to negative 70 millivolts. The change in voltage from positive 30 millivolts to negative 70 millivolts is referred to as repolarization. Because it takes time for potassium channels to close, the voltage goes below negative 70 millivolts before returning to steady state voltage at negative 70 millivolts. This is called hyperpolarization. Now let's address some important questions to better understand action potential. First, let's take a look at threshold and its importance. To initiate action potential, the membrane must depolarize from negative 70 millivolts to at least negative 55 millivolts. This critical level of depolarization is referred to as threshold. Once depolarization reaches threshold level, more sodium channels will open up, allowing action potential to reach peak at positive 30 millivolts, which is then followed by repolarization. Keep in mind that if the change is lower than negative 55 millivolts, no action potential will occur. That's why we say that action potential is all or none. It either happens or it does not. Will stronger stimulus lead to a bigger or stronger action potential? The answer is no. All action potentials peak at positive 30 millivolts. So one action potential is not bigger than the other. However, stronger stimulus will initiate multiple action potentials more quickly, but the individual signals are not bigger. After action potential is initiated, how does it propagate or move along the axon of the neuron? In response to a signal, the beginning of the axon becomes depolarized. The depolarization spreads down the axon. Meanwhile, the first part of the membrane repolarizes because sodium channels are inactivated and potassium channels have opened. Depolarization is followed by repolarization along the axon, allowing for action potential to move towards the axon terminal. Does propagation of action potential occur the same way in myelinated and unmyelinated axons? The answer is no. What has been described on previous slide occurs in unmyelinated axons. However, when myelination is present, the action potential propagates differently. Once sodium enters the cell, it moves down the axon until it reaches first node of Ranvier, where more gated sodium channels are located. They will open up due to depolarization from negative 70 millivolts to negative 55 millivolts by the initial sodium ions, and the flow continues down the axon until following node of Ranviers with voltage-gated sodium channels are approached. Myelin acts as an insulator that prevents current from leaving the axon. This increases the speed of action potential conduction. In demyelinating diseases, such as multiple sclerosis, action potential conduction slows down because current leaks from previously insulated axon areas. Can another action potential be initiated while the first action potential is still in progress? The answer is no. While the first action potential is in progress, another one cannot be initiated. This is due to absolute refractory period, during which the majority of voltage-gated sodium channels are inactivated after sodium enters the axon. As voltage-gated sodium channels transition from the inactivated to closed state, they become capable of being activated or open, and therefore membrane becomes capable of supporting second action potential. The period during which second action potential can be initiated is called relative refractory period. To produce second action potential during relative refractory period, the depolarization must be larger from the first one because the membrane is hyperpolarized. In other words, initiation of second action potential requires greater stimulus than the one that initiated first action potential. This is because potassium is still rushing out of the cell due to the first action potential, and any sodium that enters the cell will not depolarize the cell, but will only keep the cell from hyperpolarizing, unless the stimulus is stronger to move from hyperpolarized state towards -70 millivolts and then towards threshold. How neurons communicate with each other and target cells such as muscle cells. Before I get into details as to how signal moves from one cell to another, let me first explain some of the vocabulary. Presynaptic cell is the neuron which sends signal to a receiving cell. The receiving cell, whether it is another neuron or muscle cell, is referred to as postsynaptic cell. Space between presynaptic cell and postsynaptic cell is referred to as synaptic cleft. Tips of axon terminals are referred to as synaptic end bulbs. In the synaptic end bulbs, you will find small round membrane pockets known as synaptic vesicles. These synaptic vesicles are filled with molecules known as neurotransmitters. Neurotransmitters are chemical messengers used by nervous system to transmit messages between neurons or neuron and muscle cell. When an action potential arrives at the axon terminal of presynaptic neuron, it opens up ion channels that let calcium ions into the cell. Calcium causes these synaptic vesicles to fuse with presynaptic membrane, releasing their contents into the synaptic cleft. The neurotransmitter diffuses into the synaptic cleft and attach themselves to a specialized molecules known as receptors on the surface of the postsynaptic cell, whether it is another neuron or muscle cell. Receptors can then allow charged ions into the postsynaptic cell, changing its membrane potential. If enough receptors are activated, the membrane potential will eventually reach threshold, and the postsynaptic cell will generate its own action potential that will travel along the postsynaptic cell. Eventually, the neurotransmitter molecules in the cleft will diffuse away or will be broken down, thus ending that round of neurotransmission. All in all, this entire process takes a couple thousands of seconds. Different classes of neurons can make different types of neurotransmitters. And every neurotransmitter has its own specific type of receptor. Nicotinic acetylcholine receptors are ion channels which open when bound to neurotransmitter called acetylcholine and let positive ions into the cell, causing it to depolarize. This type of synapse is the one found at the neuromuscular junction, the place where neuron contacts and activates muscle cells to generate contraction. This is the end of my presentation about membrane potential, action potential, and chemical synapses. Please subscribe if you find my videos helpful and educational.